the ribosome will come also and attach into the mRNA and start to synthesize the polypeptide. So that in here, the transcription and translation is a continuous process because it happened nearly immediately at the same time. One other difference is the ribosome in prokaryotic cell is smaller. The ribosome was about 70S, which S is the unit for the centrifuge fork unit. Its name followed the scientist who invented this unit. And this 70S was composed by two subunits. One is have a size of 30S, and the other have a size of 50S. Meanwhile, in the eukaryotic cell, because in eukaryotic cell we have the nucleus membrane separate the nucleus region and all the DNA inside with the cytoplasm. So the transcription was happening inside the nucleus region, inside the nucleus membrane. And after the mRNA was synthesized and had been through to the major mRNA processing that we will discuss later, it will getting out of the nucleus region through the nucleus membrane hole. And after that, the translation starts. I mean, the ribosome will come and binding into the mRNA and start to synthesize the polypeptide. So in eukaryotic cell, the transcription and translation is not occur continuously. The translation happening after the mRNA have been going out of the nucleus region. And the size of the ribosome is larger, which is 80S, consists of two subunits. The small is 40S and the large is 60S. A little bit larger than the ribosome in prokaryotic cells. So let's go further on the transcription in both prokaryotic cell and eukaryotic cell. So in eukaryotic cell, we have a typical promoter of the prokaryotic cell is come as follow. So let's hypothesize this is the position +1. This is position +1. +1, that means that this is the position that the mRNA will start to synthesize. Okay. So before this position 10 nucleotide, we'll have the region called the Pinot box. We have the compose, the sequence TATAAT. And we have another region is at the position -35. That means 35 nucleotide from this position is the TTGACA box. So how we can, how the scientists can identify which is the favorite promoter binding region of a genes. So it's based on the motif binding. Motif binding. Okay. So in here you can see a motif binding of a transcription factor that they have sequence in so many many positions of the gene. So for example, identify motif binding transcription factor. So first, they put it in the genome. So this is the genome of organism. This is, let's say, the genome. And they let the transcription factor binding into it. So the red one here is the transcription factor, for example, it binding in the favorite region. So the red one is the after the transcription factor binding here, they break down the DNA sequence and start to sequence those sequences. So for example, if we have a motif binding here, is that because in many times they had find out that in the position of 3, here 100% is a T. Also in 4, in C here is, and the A in the position here. And the other, for example, in the position of the 9 here, 50%, they see that it's an A, and 50% they find out it's a C. So this person in this position is either A or C, or in the other case, the transcription factor will not binding. But in this position, 3, 4, 6, and 10, if the nucleotide would change, that means transcription factor will not binding in these sequences. And that's how we can identify which is the region that a transcription factor binding in. Okay. So come back to our slide. So in here we have a typical prokaryotic promoter here. We have 2 regions that favorite for the transcription factor binding, the Pinot box and the -35 box. The typical eukaryotic promoter is from the TATA box, which is in the -35, sorry, 25 of the transcription starting site. CAAT box in -80, and one region really interesting is called the GC island. So GC island is a region that contains many G and C nucleotides. And this GC island is related to the DNA methylation that we will discuss further on chapter 7 in the epigenetic chapter. Just for now, just remember that if the gene was mostly DNA methylated, that means the genes will be turned off during transcription, mostly. So the state of transcription start when the transcription factor among those DNA polymerase will come and binding in the region and start to synthesize the RNA polymerase. And it will start synthesize the mRNA along of the length of the gene until it gets to the termination. Then it will release the DNA template and also the RNA product out and finish the transcription process.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex, able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors called coactivators link the basal factors with a third set of transcription factor proteins called activators. The activator proteins are not shown.
So come to a very important term. What is template encoding strand? So different with DNA replication, which is happen in both strands of the DNA template. The transcription happen only in one template. So in here, the same with the DNA polymerase, the RNA polymerase only working on a polymerase only working from 5 prime to 3 prime. Okay, so as the consequences, first it will binding into the other strand. This strand here, the 3 prime to 5 prime here. So the RNA polymerase will binding in here and start to synthesize the mRNA. So because the transcription factor binding in this template, so we call it the template strand, because the transcription factor use it as a template. And following the complementary Chagat rule, we will have A will link to U. So the second is the new mRNA will be U binding to A, C binding to G, A is binding to T, G is binding to C, and so on. So this sequence, the sequence of this will have the same sequence like the other strand of DNA. So the sequence of this mRNA will have the same sequence as this strand. So replace only the T by the U, all other sequences are the same. So that is why we call it the DNA coding strand, because the mRNA have the same sequence with this DNA strand. And sometimes we call it also the positive strand, and the other, the template strand, is called the negative strand. Okay. So I have made a small table here to compare between the differences between replication and transcription. So in DNA replication, we have template is double strand. Both strand is a template, and in transcription is a single strand. Only one strand is used as template. And as a result, it creates a double strand DNA in replication, but only one strand of RNA in transcription. Okay. The substrate, of course, this product is DNA, so this is delta nucleotide. TNTP, TP is stand for 346, and in here is the ribose nucleotide 346. Okay. In DNA replication, it requires RNA primer, but in transcription, it requires no primer. So you can see it here. There's no primer in here. The RNA polymerase can synthesize directly from the DNA template without any primer. And the enzyme one is DNA polymerase to provide DNA, and the other is RNA polymerase. Okay. So I think today's lecture is finished here. The next part on the gene structure of the prokaryotic cell and the eukaryotic cell, we will discuss about it in Monday. Just one thing before you sign up this class is that in the Moodle account, so I have updated the slide for the chapter 1. So if you want to keep your slide update to the lecture, please download the new version of chapter 1. And about the other chapter, I will keep being updated after I finish each chapter. Okay. Okay. Do you have any